So good morning, everyone. Today we have Professor Andrei Lupas as our seminar speaker. And but before starting, so David Ruano is going to introduce our speaker. So David, whenever you're ready.
So hello, everyone, and welcome Andrei Lupas to our seminar series. And thank you very much for accepting our invitation. Dr. Lupas, after getting his degree in biology in Munich, he moved to Princeton, where he started studying the structure of proteins. He got his PhD, moved back to Germany in 1992 to the Max Planck Institute for Biochemistry, and fourth to SmithKline Pharmaceuticals. Since 2001, he is director of the Max Planck Institute for Developmental Biology. Dr. Lupas is an expert in structural biology, where he connects to the origin of life. He analyzes and describes the basic motifs common to the structure of proteins with diverse functions. These motifs may have arisen in the prebiotic world by coevolution with other molecules like RNAs. Indeed, combining and repeating these motifs allows a likely reproduction of how fully functional proteins emerged in the beginning of life. Thanks again for accepting our invitation, and I'm sure everyone will enjoy your talk. So,
thank you very much. So let me go in medias res, if I can. It doesn't allow me to move. That's so odd.
It doesn't allow you to go to the first slide?
Now it does. For some reason, it was hung up. Okay. All right. So
we don't see your first slide. We just we see the one with the animations. I think the one that you mentioned that you have animations.
You don't see the origin of life, a case of spontaneous generation?
No, sorry. No. I'm very sorry for this. I apologize.
Let me then. Okay. Now you can see it. Right. So
yes,
we'll
yes,
do it this
go
way,
ahead.
Rather than trying to figure out what's happening. Allow me to resize this. Okay. All right. So in essence, I have tried to summarize on the slide the history of the Earth, shown in black letters above the timeline, and the history of life, shown in blue letters beneath. This is, of course, extremely selective. My main points are, one, that the origin of life took an extremely short time relative to the total timeline. So it's been an extremely efficient process, a staggeringly efficient process if we think of the complexity that the last common ancestor must already have had. By the time of the last common ancestor, we had DNA, RNA, and proteins. The prototypes for all the main proteins had evolved. All the core biochemical reactions had been encountered and established. The core metabolism was set. Translation, transcription, all these extraordinarily complex processes had already evolved and were established. So what we see here is that from the point where we have liquid water to the last common ancestor, we had less than 1 billion years, probably much less than 1 billion years. So this is one of my points. The origin of life was a very efficient chemical process. The other point I would like to make, and that is more diffuse over this timeline, is that life progresses by catastrophes. And the bigger the catastrophe, the bigger the progress in life forms. So let us look at some of these catastrophes. For example, the massive ice ages, which are also known as snowball Earth episodes, where essentially the entire surface water on the Earth was frozen. And these coincide with the origin of eukaryotes and with the origin of multicellularity. And in both cases, we saw substantial increases in the level of free oxygen available to organisms. And to me as a biologist, these are directly connected. Eukaryotes arose because they needed to deal with oxygen, and only bacteria knew how to deal with oxygen. And so this was the origin of mitochondria, of the symbiosis, because before this time there were the archaea and the bacteria, and probably an ancient archaeon took up a bacterium, formed a new symbiosis, and that was the eukaryote. And then here at multicellularity, once we had enough oxygen, it was clear that you could now sustain body plans that were sufficiently large to allow multicellularity. You couldn't do it with a lower energy source. And of course, you couldn't do it without ozone, which protected the increasingly growing genomes from radiation from space. All right, we could talk a lot about this, but I would just like to take this point to emphasize how efficient this process was and to look at what must have happened in that process. And now, yeah. And it must have started with what I would call metabolic networks. So networks of reactions which allowed the emergence of more complex molecules from the simple molecules, gaseous molecules at the beginning, carbon monoxide, ammonia, H2S, H2, and these metabolic networks allowed the emergence of what turned out to be building blocks, like the amino acids, or like various sugars, like the bases that form the core of nucleic acids. And at some point, these also started to form polymers. Now, for amino acids, that has already been studied quite extensively, and it's fairly well established. For other molecules, it's much more complex, how polymers could have arisen. And in my opinion, the question of how the first nucleic acids arose is still almost completely open. People can make polymers of nucleic acids if they are given the building blocks, but the building blocks are sufficiently complex that it is a real challenge to figure out how substantial amounts of them could have emerged. Now, all of these steps must have happened in what I called here the pre-RNA world, the part of chemical evolution in which we did not have real nucleic acids. But at some point we did have nucleic acids, and there are many arguments to support the notion that whatever the first polymers were, the ones that led to the emergence of life were built on RNA. And that's why I wrote here on the side, the RNA-peptide world. Do you see my cursor? Yes, okay, good. And at some point, these polymer networks would have encountered the ability to reproduce autocatalytically. And as I will explain to you, this is, in my opinion, the beginning of life. So we have further steps that were really important: compartmentalization, namely the ability to maintain the products of a metabolic network within a confined space, emergence of a code that provided the interdependence between the polymers forming life, and at some point also equal cell division, which allowed the complete transmission of information from parents to, from mothers to daughters, essentially. All right, so why do I say RNA must have come at the origin of this? And the answer goes back to the 1970s, when researchers, mostly physicists actually, who were working in biology, realized that in all biological entities, information flowed from DNA through RNA to protein. And then proteins were the agents which performed essentially the entire chemistry. And indeed, in our world today, Nucleic acids are only there to specify which proteins are made when, in what amounts, and everything else is done by protein, including the replication of the information, its transcription, translation. All these things are done by protein, with the exception of this huge RNA-peptide complex, the ribosome, which is the factory that actually concatenates the amino acids into polypeptide chains and allows proteins to happen. And so already in the 70s came the first proposals that the original world was an RNA world, because the RNA was taking the center stage in the conversion of information to chemical capability. And what people, of course, observed was that the RNA could also encode genetic information, but it could also catalyze reactions, most prominently, as we saw, the ribosome, which makes proteins. However, even though the RNA can do coding and catalysis, it is substantially limited in both these activities. And to my knowledge, no RNA genome has been evolved that can have more than about 30,000 letters. So that seems to be an upper limit. Right, and when I started on these ideas almost 30 years ago, it became clear to me that peptides were offering a very large number of benefits to RNA molecule, including the ability to chelate various molecules essential for the reactions in these metabolic networks, to promote the thermostability and folding of RNA, to promote oligomerization, support adhesion to abiotic surfaces. And also, I think this is becoming more and more important in my thinking. Peptides, many peptides are membrane active. If we had an envelope that allowed to keep the products of a metabolic network in a confined space, it would at some point have had to be lipids. And peptides interact very favorably both with nucleic acids and with lipids. So they would have formed a natural bridge between the nucleic acids and the lipids, whereas nucleic acids and lipids do not really go together very well. And so I would offer here my claim that the RNA world was in fact an RNA peptide world. Peptides were there from the beginning, and they were essential for the emergence of autocatalysis. I will not explain this further for reasons of time, but I would be absolutely happy to discuss with you. Basically, let me get to what I consider to be the origin of life. The polymer networks that led to autocatalysis were RNA peptide networks. And they were the first to embody the 4 criteria that you need for Darwinian evolution. These are encoded, let's say, in the 2 statements that you are, I'm sure, very familiar with: descent with modification and survival of the fittest. Descent means that the system can replicate. With modification means that the replicates are not identical to the parent and they're not identical to each other. So there's an emerging variation. Survival of the fittest. Survival means not all of them go on to replicate themselves. And the fittest, well, that's an interesting point, because biologists cannot really tell you what fitness is. Biologists measure fitness by survival, right? Which is you survive because you are fit, but you measure fitness by survival. It's a circular situation. Darwin was absolutely not favorable to the statement. He didn't invent it, and he really disliked it and wrote against it. But the point is, I don't care what fitness is. All I care is that the survival is not random, right? And whatever you use to define fitness, it is clear that it is not a random process, which means you have to have offspring. The offspring cannot be identical to you and to each others. You're going to select a subset of these offspring, and you are not going to select these randomly. And once you have these conditions in place, the whole system will have a directionality. It will move towards your selective pressure, whatever pressure you apply, right? And so this is the beginning of life. And so if we go to Darwin's statement at the end of The Origin of Species, whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful have been and are being evolved. And the point is, if I'm correct, and as shown in Escher's drawing, proteins make nucleic acids and nucleic acids make proteins, and this circle closing gave us the first autocatalysis, then the beginning was even much more simple than Darwin imagined. But I think that in the end, this will be the answer. We need to find networks of peptides and nucleic acids which can autocatalytically replicate from a starting condition which could have been arrived at by abiotic processes. All right, but I don't know, maybe I won't even live to see anyone use this to make life in the lab, but I'm sure it will work. All right, so then we have from here the transition to the DNA protein world, right? The world of RNA and peptide essentially relied on RNA being a structured scaffold on which peptides formed cofactors by adhesion to the nucleic acid. Whereas in the world we see today, we see proteins as autonomous agents, independently folded, capable of catalysis without nucleic acids, capable of essentially doing the chemistry of life. And we see DNA as a molecule that essentially is a pure coding molecule. This is where the information is stored, but DNA does not do catalysis. It's just the information repository. So we see a complete split of the roles, and we see a marginalization of RNA as a mediator between DNA and protein. And the question is, why did this happen? If the RNA world was functioning, biology does not progress by leaps and bounds. Biology progresses from step to step because each intermediate has to survive and have offspring, else the chain is broken. And so you cannot say, oh, it would be so lovely if we suddenly use DNA, because at first it won't work very well, and you will lose the race for resources for life. So I would suspect that this was another one of these instances where a major catastrophe forced life to essentially restructure around new molecules. And I'm particularly attracted to the so-called late veneer, which I heard about from Steve Moises, and which I understand is by now something that has been discussed extensively in geology, geochemistry, and earth sciences. The idea that the heavy meteorite bombardment ended with a major impact, which essentially brought a lot of the heavy elements to the surface of the Earth, to the crust, and also caused a major spike in temperature. And RNA is by far the least stable of the macromolecules. So organisms which eked a marginal existence with DNA as the information repository, for example, because they were defending themselves, they found this as a mechanism to defend themselves against viruses, these organisms suddenly were the only ones surviving. So even if they formed only a minute fraction of all living systems, if you kill all the other systems, then by necessity, these ones will survive, no matter how incomplete or how difficult their survival would be. If they're the only ones, it doesn't matter. And so the idea is that a major catastrophe wiped out the RNA world. And the only function that really survived in the RNA is the function of making protein in the ribosome. So it's a speculation, but somehow as a biologist, I need an explanation why the RNA world was pushed to extinction. Right, but whichever way this turns out to be, this is not my research. I'm specifically interested in one major hurdle that life faced when RNA went away, and that is called the protein folding problem. It turns out that if you accept what I told you about the RNA-peptide world, the peptides did not have a structure of their own independently of the RNA. They became structured in context of the RNA. But then when the RNA went away, how did they become structured? And if they don't become structured, how do they do chemistry? You can't do chemistry without structure. And so this was the question which I wanted to address in starting my department here in Tübingen. And I will try to explain to you my thoughts and what came out of it. So I started from the observation that present-day life results from the chemical activity of proteins, and proteins have to have a three-dimensional structure to provide this chemical activity. There are natively unstructured proteins. They're fairly recent in evolution. They're mostly confined to eukaryotes, and they have a completely different origin, which again, I would be absolutely happy to talk to you about, but I won't do it here. Now, in contrast to nucleic acids, which fold spontaneously, proteins do not form spontaneously. So if you've ever tried to purify a protein after expression, you will find that it mostly sticks to surfaces. It's difficult to get it on the purification column. It's even more difficult to get it off the column. And when you get it off, very often it does not have structure. So you have to do a refolding step in which you get it to reassume its structure. Very often this process is not efficient, and a lot of your protein will end up in aggregates. Now, you could say, well, that's because we're ignorant of how nature does it, and we're just not very good at it. But the answer is we're actually pretty darn good about it. The point is nature is not very good about proteins because natural proteins are not particularly stable. So if you think of having a fever above 42 degrees, more than a few hours will damage you, more than about 48 hours will kill you. And in between, you will suffer significant brain damage to the point where you would be turned into a vegetable, right? So 48 hours at 5 degrees above our temperature optimum is enough to kill us, right? And the reason it kills us is because our proteins quit service. They are optimized to work at 37 degrees. And if we don't give them 37, they start quitting very quickly. And we know that also from degenerative diseases like Parkinson's, like Huntington's chorea, like Alzheimer's disease, cystic fibrosis. These are all degenerative diseases in which one or a few proteins quit their service and do not fold anymore. And then we die at some point. Other proteins, we only get sick from proteins that are not important. At first, I thought that was so counterintuitive, but it's true. The important ones, you're dead almost immediately, right? If one of your important proteins quits, you're gone within hours. Yeah? So basically, our proteins are not very stable, and protein folding continues to be a major issue in all life forms. When I was a postdoc with Wolfgang Baumeister in Munich, we were studying folding factors, and the way we isolated a folding factor from an archaeon was by heating the archaeon, which had a temperature optimum of 104 degrees to 110 degrees. And before it died, 70% of the cell mass was this one chaperone. The cell was not doing anything but trying to save itself, right? So heat shock is just relative to what your optimum is. But once you are above the optimum, you won't last long. All right, so proteins have this tremendous problem. How did they originate? Folding is such a huge problem. How did it arise at the end of the RNA? How did we go from peptides to folded proteins? And the way I envisaged this was that we had building blocks, pre-optimized chains of peptide size, which could assume the so-called secondary structure elements, alpha helices, beta strands, through interaction with the RNA. And they evolved to be able to exclude water with a scaffold in order to assume these structures. And so when the RNA went away, if you took these building blocks and put them together into longer chains, they would be able to exclude water between each other and become independent of the scaffold, which is not there anymore. And so the way I envisaged this happening is that we had this pre-optimized set of peptides, and they got arrayed into these complex chains, which were able to assume folding, and with folding also able to assume catalysis. And so I figured that if this was true, we should still see the traces of these ancient peptides by comparing modern proteins. And the way I thought about it was through language. I've always been fascinated with language. I grew up in Romania in an environment where we were speaking Romanian, French, German, English, and I noticed similarities and dissimilarities, especially with Hungarian. I'm from Transylvania. So I would hear Hungarians and they sounded so completely different from other people that it occurred to me that some languages are more similar to each other, like Romanian and French, at intermediate distance, Romanian and German, or really far away, like Romanian and Hungarian. And so here I show you an evolutionary tree of a word which illustrates the kind of processes that proteins undergo. Let me start here with this root, kane, which we know from Babylonian cuneiform tablets. So it goes back a long way. It means a reed, the swamp plant, the reed. It gave descendants in all Semitic languages, also in Arabic and Hebrew. The Greek took this by acquisition, by lateral transfer from local populations when they moved from the North Caucasus into the Peloponnese. And they used the word as kana. Now, between kane and kana, the distance is not very large. Now, for reasons that I completely fail to understand, the Latins took the Greek word as kana. Now, Rome was founded in a swamp. The Romans must have had words for reeds. Why they stopped using them and took the Greek word, I don't know, but then I'm not a linguist. Fact is, the Latins used kana for reed, and via the French can, we arrive to the English cane, which is very similar to its root 5,000 years ago and still means the same thing. It means this swampland, plus a few other things. So let's look at this plus few other things. What happened? And here we see a number of paralogies. Biologists call orthology the linear descent. I'm the ortholog of my father. But paralogy, the multiplication within the same generation. So I'm the paralog of my sisters. They are all orthologous to my parents, right? But they are paralogs of me and to each other. All right, so what the Greek did is they took this word and used it to denote rulers. So the reeds have the advantage that they're really straight. They have nice nodes at intervals, and they can be used to measure things. And so the Greek made rulers out of them and used the word kanon to denote this object, and also to denote the rule, the abstract concept of this rule. And the Latins took it as canon, and this gave us the canon as the church rule. The church canon is the set of rules of the church. And it also gave us the musical form. When you sing a canon, you sing by rules. So it doesn't come from the Latin canere, to sing. It comes from rule, from canon. All right, another paralogy in Greek times, kanastron. The Greek used the flexibility of young reeds in order to weave baskets, and they called these baskets kanastron. I believe there is a Spanish word which is similar to that. Canasta, in any case, is a game which originated in Spain and is, I think, throwing cards into a basket. Right. And the Latins came up with another paralogy. They made pipes. So they noticed that the reeds are hollow. If you puncture the membranes, you have a complete tube. And they are tapered, which means one end is fatter than the other end. So you can take them and just push them into each other and make long tubes, which can be used to transport water. And that's what they did. And they called them canales. And from this comes the channel and the canal and the canyon, and also all these other derivations like cannelloni and cannelure and cannula, and the can. The can does not come from the canister. The can is an independent evolution from the root cane. And so what we see here is that in essence, through time, while we preserved mostly the aspect of the word, we have changed its meaning in very substantial ways so that the last common ancestor of a church rule and of a TV channel lies in a Sumerian plant. Okay? And this is exactly how I envisage the evolution of proteins. This is what happened. And I've summarized it here in this slide. Let us start with this fragment here, which is a primordial fragment that I will show you presently. We think it evolved in the context of RNA. And we certainly see it in a protein that occurs in the ribosome. This is the last survivor of the RNA world. And interestingly enough, this protein L27 cannot fold without the RNA. So it looks like a protein, but it's not an autonomous protein. It requires the RNA to fold. But it gave offspring, namely, for example, this exosome subunit here, which is autonomously folded and does not require RNA to fold. And so I see this as a chain illustrating the way I think about this. First, we had a peptide which absolutely required RNA. Then the peptide got much more complex and came to the edge of becoming autonomous. And then the RNA went away and the peptide became fully autonomous. Right. I won't go to all these paralogies, but they can be explained in the same way in which I explained the evolution of the word cane. And that's why I made these two figures. They were side by side in the paper we published on these fragments. So now let's see what we did. We basically decided that the similarity in sequence was the key element. The structure was a derived feature. And so we built tools in order to detect more and more distant but statistically significant similarity in sequences. This tool is by now fully established as the most performant tool for deep homology. It was very fortunate for us that we didn't have to argue all the time about why we're doing it. And we made this public. Starting in 2005, we put up not only this tool, but the entire toolkit of bioinformatics, which is very widely used. We typically answer about 500,000 queries a year to the toolkit. And with this toolkit, we compared proteins of today, right? The idea is that if the fragments are older than the proteins, then the fragments could have been elaborated in different ways. So the proteins will not be similar except in the fragment. The fragment would be the only thing similar between proteins. And so here you see this fragment, the helix hairpin helix motif. I've marked it red in all these proteins. Here you see a superposition. You see the fragment is very well preserved. It has not changed much in 3.5 billion years. And its sequence is also quite recognizably still related. But the rest of the proteins shown in gray, you see, is completely different. Each one of them took a different path to becoming a protein. All right, and we did this systematically over proteins of known structure, and we came up with this vocabulary of peptides which predated the origin of proteins. And we published this in 2015. By now, I read in the introductions of people in my field that, as is generally known, proteins started as peptides. And then they often don't even cite us because, of course, it's generally known. But to me, this is extremely deep satisfaction because for 20 years, I went around conferences explaining these ideas before we actually had evidence. And people would say, oh, you know, it's just speculation, there's nothing behind it. All right, so let me show you why people have stopped calling this speculation. And the answer is because we don't just assemble a vocabulary. We actually work in the lab with these peptides to show how they could have produced folded proteins by simple genetic operations. Anything complex would not fit. We need something that's simple and extremely likely to have happened. And so allow me to take this fragment here. It's 4 beta strands connected to each other. This is one of our larger peptides at about 35 to 40 amino acids. And it is found here. You see, this is the fragment, and it gave us, for example, proteins that look like this. These proteins are called propellers because the fragments are arranged like the blades of a propeller. The whole thing looks like a propeller, but they also form prisms where the fragment forms the 3 sides of the prism. And they also form structures which are neither prisms nor propellers. In fact, these structures don't really look very repetitive, but they are. All right, so what did we do with this? What we did is we suspected that nature continues to make folding proteins from this piece, from this fragment. And so we looked in nature for folded proteins which arose by amplification of such a fragment. And we looked for the proteins that had been amplified the most recently, where we wouldn't have to explain a lot of things, where all biologists would say, yeah, of course, that was amplified yesterday. And this is an example. So here we see our fragment repeated 14 times in a protein. Right? There is here a process of shuffling a piece to the other end. It's called circular permutation. I won't go into it. It's well known to protein evolutionists. And I will focus on this repeated unit. Now, this stuff makes this structure. This is the crystal structure. It's not a model. This is the crystal structure of this repeat. Okay? And we took this blade here, the one that's marked in red, and we studied whether we could make all sorts of amplified forms, not just the one that nature has made. And in this, we were aided by the fact that the DNA encoding this has practically no mutations that are called synonymous, which means mutations that specify the same amino acid. So the genetic code is redundant. Some amino acids are coded by more than one codon. But here we find that all the changes in the DNA, which I've marked yellow, are non-synonymous. They cause a change in the amino acid. So a biologist would tell you that this is practically impossible. You can't have all these non-synonymous mutations and not a single synonymous mutation. And the answer is it is possible, but it means essentially that this protein evolved yesterday and is under infinite evolutionary selection, which is of course untrue. We're just approaching a boundary condition. In fact, it probably evolved in the last 100 years and the selection pressure is large, but certainly not infinite. All right, but let us look at what we did with this. We took this fragment and we made a construct seen up here, which has two blades in a row. We were unable to fold the single blade, but as soon as we put two blades together, we got this 8-bladed propeller, right? This is a crystal structure. Everything I'm going to show you is crystal structures. So there's no doubt that this is what this construct does. So as soon as you put two blades together, you start folding. Now, repetition is a very, very cheap and simple genetic operation. So it's obviously extremely easy for a blade to become structured. It's right at the edge of being structured. All we need to do is duplicate it. So then we triplicate it, right? You see here that now we have 3 blades together, and you see that it makes a 9-bladed propeller, right? With 3, you didn't expect it to make 8. All right, now keep in mind the parent has 7 blades, right? This propeller here has 7 blades. There's 14 blades overall, and they make 2 7-bladed propellers. So the parent has 7 blades. This one has 8. This one has 9. Now we put 4 blades in a row here, and now you see that you get 8 again, but it looks different from the other 8. Let's not go into more detail. An interesting one is this one. Unfortunately, the animation is gone, so you can't see it other than from the side. This is what I would call a helpful monster. It's something that's not a propeller. The 2 halves, even though they are the same, each half has 5 blades. Only one makes 5 blades, the other one makes only 4. One blade turns into a helix. The 2 halves turn anti-parallel, so it's not a propeller. It's a donut, but not a propeller. And the hole that arises is filled by the helix. So it refolds substantially. So we see there's a lot of folding potential. There's a lot of variability. The blade is really close to the ability to make all sorts of things. You just need to give it simple genetic events to get it to fold. All right, I'm actually coming almost over time, but I do want to show you this element. This is another one we did. Right, you see here, this is called a helical hairpin. There's 2 helices arrayed in a way that looks like a hairpin. And it's in the ribosome. It's shown here in red. And in essence, what we observed was that folding progressed from the inside of the ribosome to the outside, almost as if you saw slices of an onion growing towards more and more folding as the ribosome became larger. And so in the core, you don't see any folding almost at all. You go a bit out, you start seeing folded structures. You go out further, now there's real structure accumulating. And at the edge, there are full proteins that are partly even independent of the RNA. I've not shown the RNA in this picture. You only see the peptides. The RNA, of course, is there, but you wouldn't see much if I left it in. And so we imagine in this explanation I gave you, we imagine that folding progressed from the inside of the ribosome over time to the outside. And then it was just one step from the edge of the ribosome into full independence of the polypeptides. And so collectively, in our opinion, the structure of the ribosome chart a path of emancipation of polypeptides from the RNA scaffold, giving us all the folded proteins that now populate our cells. And so to return to this fragment, in essence, we wanted to make this piece here with this piece, right? We wanted to recreate this step. And so we took, now we won't go into that. We took this hairpin, which is in the ribosomal protein, and we tried to make this structure by repeating the hairpin 3 times. Right here, you see the superposition of this yellow part with this blue part. You see they're quite similar. So we made the construct. The model that we built is in gray. The crystal structure is in cyan. And you see we pretty much got what we thought we would get. So it's enough to take the fragment, make 3 copies of it, and you get your protein. And this piece folds by itself. So the fragment from the ribosome is really close to the ability to fold. You just need to give it one genetic event that will get it there, right? And so I have quite a few more examples. This is why biologists have listened to us, is because we do the experiments and we show that it works. And everybody in biology will say, oh, that's so simple. I didn't believe this could happen, but it happens. And it doesn't happen only in these 2 instances. We've shown it in one instance after another. And others by now have too. So it's clear that if you work from pre-optimized fragments, folding is really close. You don't need to go far to achieve folding. And so this idea that the RNA world would have pre-optimized the fragments up to the point where they could start folding is becoming really substantial. Virtually everybody now in biochemistry accepts that this is true. All right, let me end with the following images. What I've shown you is simply evidence of plausibility. What I've shown you is that the processes that can lead to structure are simple. But whether this actually happened this way, we have no idea. We cannot travel back in time, right? So unlike physicists, we cannot do experiments to probe the ancient times. All we can do is create systems where we replicate the effects and say, if it works now, it should have worked then. Okay, and I compare this to this little game, which you certainly know if you have children. If you connect the dots according to the numbers, then you get a nice picture, right? The problem for us is we don't get the numbers. So how do we know what to connect? And even worse than that, most dots are extinct. So the pictures we recreate may or may not be close to the original image, okay? We are absolutely aware of that, and I would never claim that I'm recreating life. All I'm doing is I'm showing that it could have worked with a bunch of simple principles. And with this, I would like to thank the people who over the years were the main agents of what I have shown you. And I would be happy to take any questions you have.
Thank you very much, Professor Lupas, for this very nice and very interesting talk. So maybe we can move on to the questions. Are there any questions?
Should I unshare or do we need?
No, maybe just leave the slides just in case we need to go back to any of them. Maybe I can start the questions, if I may, or maybe Victor, maybe you can. We can't hear you.
Okay, now?
Yes.
Okay. Thank you very much, Andrei. I enjoyed a lot your talk, and your presentation is amazing, the work you have done. Yeah. So I have the impression then that here at the end, I would say that the role of the RNA world is more simpler than perhaps previously thought, in the sense that I mean that the RNA somehow is a helper for the protein world somehow. So the protein somehow, takes use the RNA just to say, okay, I need you to stabilize, I need you to make the structure, etc. So I think this what I extract from your talk. So is this the idea? And also, I wanted to ask you do you think that this interaction with peptides, with nucleic acid, either RNA or DNA, may have been one of the main responsible for example, the enantiomeric selection of L peptides rather than D peptides, for example?
Yes. Yes. So basically, if we look at this image, an aspect that I did not mention, but that is biologically true, is that the only molecule that can make protein is RNA. And the only molecule that can make RNA is protein. The polymerases do not have cofactors. They do not have any RNA involved, any nucleic acid, any nucleotide, other than as reactants, which are chained together. The polymerases are pure protein, right? And to me in biology, you have these very old dependencies which simply don't change because each step has to survive and has to work. And so if I see this, I'm bound to say that the ancient polymerase, the first polymerase, was peptide only. And that is why I feel so confident about the cycle closing. The peptides were making the nucleic acids were making the peptides. And the whole process only worked because peptides could assume a cofactor role in catalysis once they had the nucleic acids. Now, you say enantiomeric excess, and I say absolutely true. That's the original active site. If the amino acids are made abiotically, you'll make racemic mixtures. And chemists are spending enormous amounts of time trying to explain how a system may become biased abiotically. And I think the answer is no, it's much simpler. The RNA reaction center was selecting amino acids of the L form. And the peptides, which were ligating the bases, they were selecting the correct sugar enantiomer in order to make the polynucleotide, the nucleic acid. So I think the origin of stereoselectivity lies in catalysis.
Yeah, but is there any physical constraint that really say, okay, we only work with L but not with D? Which could be the reason for that? Why?
Random. Random.
Yeah. Mm-hmm.
Right. So one thing that I started talking about since I've started talking to astrophysicists in the last couple of years is that there are situations in which an outcome is extremely unlikely, but a functional outcome is extremely likely. It's just that there are thousands of possible outcomes and any of them would do, and you can build on it. And that's essentially the way the exact sciences, physics and chemistry work. They are probabilistic outcomes. Biology is a contingency science. Everything in biology is contingent on the situation of the state of the system before whatever happened. And once something has happened, its likelihood is irrelevant. Doesn't matter how unlikely it was. Once it has happened, it becomes the new default state for the system. And so I think there are miracles in biology, some of which are not true miracles, because any solution would have done it. So almost like playing a lottery, right? It doesn't matter who wins the lottery, someone will win the lottery. Whereas there are probably some true miracles, some events that were very unlikely, and without them, life could simply not have progressed to the next step. And in my opinion, the emergence of the ribosome, the peptidyl center, that was a miracle. Very unlikely.
Yeah.
Yeah.
I know.
But once you had it, life, complexity of life moved extremely quickly. Mm-hmm. So would there have been other peptidyl centers arising? I don't know, but we'll know at some point because we'll do the experiments.
Okay. Okay, thank you very much. Well, just the last one, because I have a lot of questions, but just have you seen or have you observed any bias in the amino acid composition in these, let's say, ancient peptides in your case? I mean, is there any bias, for example, if we are talking about the first peptide in life, any bias to those amino acids present in the meteorites or not? It's just a random sequence too.
Right. So this has become a really important question in prebiotic biochemistry, so to speak, and there's a lot of groups working on it now. Statistically, we don't see a signal. So the more recent amino acids like arginine, phenylalanine are not disfavored relative to their completely random occurrence in proteins overall. In fact, you could say arginine is favored in the ribosome because it mediates all these interactions to nucleic acids. Whereas anciently, it must not have been there very often because there was nothing to make it. It could have been ornithine, but I don't believe that. I think the interaction between peptides and RNA was mediated differently. But this will be the subject of experimentation. In any case, we don't see a signal in statistical analyses.
Okay, thank you very much.
Thank you. Are there any other questions?
Yes, I have a question. Thank you, Andrei, for your nice talk. It was very interesting and suggests many ideas. Lets you talk for a long time. But I was wondering so many things, but perhaps an interesting question is like when you talk about how the RNA world becomes the DNA world. So I was thinking about maybe there is a role of RNases there. There were proteins at some time or peptides degrading the RNA. That may be one question for you. And also related to that, so at this in this set of events we could call, so where do you think the viruses came like along?
Yeah, exactly. I think that's by far the more relevant question. I think viruses are as old as autocatalytic networks. So as soon as you had networks, self-replicating networks of nucleic acids and peptides, you would have parasites which essentially evolved out of these networks in order to prey on the networks and use them for their own replication. So I think viruses predate life, if you wish. They came right at the beginning. Parasites arise immediately in every evolutionary situation. Cheaters, right? So I think we had viruses all this time. No matter how simple we were, we had viruses. And that's why I believe Patrick Forterre, when he discusses the emergence of DNA in the context of viruses, you could think that viruses invented DNA in order to subvert the defense mechanisms of RNA organisms. To me, it seems more plausible the other way. Organisms invented DNA in order to be able to be more aggressive against RNA information. But whichever way it works, I think the origin of the first networks incorporating DNA came as a response against predation.
Thank you. So, yes, I have many questions, but it's all right. And other people can ask.
I have a lot of questions too.
Okay. Are there other questions? I have one, but it may be a very silly question. So I was wondering thinking from the physics point of view. So does the folding help in minimizing the energy of the structure? So I was wondering why these structures start to fold. Do you think it's something related with that it optimizes the energy?
Yes, so it is absolutely the free energy of the system is the driving force for folding. But beyond that, there's very little agreement what actually is happening. So most biochemists will agree that there's a hydrophobic annealing happening, that the amino acids which are hydrophobic will tend to go towards the center, excluding water from the center. And we know that you can't easily form stable, or it's very difficult to form stable peptide secondary structures if you don't exclude water. That's been documented very well. And so the idea is that the two processes go hand in hand. You have this hydrophobic collapse. It excludes water. You start forming secondary structures, and they anneal further and further into a fold. Now, I'm not sure I believe this. We are trying to fold a protein which has no hydrophobic contacts. We're really close. So I'm not so sure how important hydrophobicity is.
Okay.
And so I think the original peptides which folded on the RNA were using many processes which we are trying to recreate with our computational protein. Namely to form specific interactions which allow water to be excluded, even though the amino acids are not per se hydrophobic. For example, by making disulfide bonds, by arraying ions into the core, doing all these things. And so I think that originally when peptides interacted with nucleic acids in the absence of positively charged amino acids, which came very late, I think they were doing this through ions. So aspartate and glutamate, especially aspartate is really old, was able to mount ions, which then would form specific and high affinity interactions with the RNA. It's just a guess. But folding is still a major problem, right? And the biophysics of folding are not well understood. It's not even clear that folded proteins are in the global energy minimum of the polypeptide chain. That's more a dogma than really proven. In fact, a man named Chris Dobson has spent, well, he's dead now, spent his whole career showing that he can make amyloid aggregates from any polypeptide chain. Just give him enough time and chemistry and he can make any chain to become amyloid. And once it's amyloid, it's so stable you can't take it apart. And that means that the global energy minimum for a polypeptide chain, any polypeptide chain, is amyloid. But in nature, we don't see much amyloid, which means that maybe natural proteins are not in the global energy minimum. It's complex.
Yeah, I see. Okay.
Yeah.
And then I have another question, if I may.
Yeah.
So for these original structures, so
Yes.
what is the minimum or the let's say the smallest that you need to start seeing this folding?
Well, I can only talk about the smallest that we have used. For the others, we don't know until we show it. But the smallest entity that we have gotten to form a stable fold was 18 to 20 amino acids. We had several variants. So I would say the minimum was 18 residues.
Okay.
It's not very large.
For us chemists, we don't even see amino acids in
Well,
space. That's something that we are looking for. Yeah.
Well, that's why in this beginning slide, right, it's such an important point for me, how quickly we went from gases to something that's biochemically modern.
Yeah.
The last common ancestor would immediately recognize as a modern living being. It took, I don't know, 500 million years or so. It is amazing.
Yes, yeah.
In fact, it almost makes you believe in God.
Okay, thank you very much. Okay, so if there are no other questions, then thank you, Professor Lupas, for joining us today.
Excuse me, just one question for Andrei. What do you think about the possibility that this, let's say 500 million or even less, period of this amazing chemistry happened in another planet, for example, on Mars? Do you think this could be feasible? Could have been feasible?
Yes, I think everything up to the first miracle would happen everywhere you had the chemical conditions for it. Now, the question is, was there a miracle? And if it was, how likely or unlikely was it? So how many of these protoplanets do you have to get starting with the right chemistry in order to get to something that goes beyond simple metabolism?
Mm-hmm. Can you imagine, for example, we are here very familiar in our institute talking about the protoplanetary disks and this type of things. And could you imagine that, for example, the true reactor of these simple and primitive peptides could have been also in these protoplanetary, let's say, bodies, that different sizes, etc.
Peptides, yes. I think it's really cheap and fast to make peptides, but there are, of course, most of them are dipeptides with just 2 amino acids stuck to each other. Mm-hmm. Making peptides of a length that becomes relevant would be considerably more unlikely because it's an exponential decay of the likelihood. And the main problem I see is nucleic acids. I don't know how you get nucleic acids. That's a huge problem for me.
Nor even RNA?
No, I mean, the problem is, okay, I can follow John Sutherland and Matthew Powner in their schemas to make nucleotides. But how do you connect the nucleotides? There's no polymerase. All the polymerases that have been made are either protein or if you take the ones from the guy in Cambridge, they're RNA, but they're really complex. They're huge things. Whereas what I'm looking for is short peptides, fairly short peptides, which have the capability of annealing bases, nucleotides together into the first chains. So I think the peptides started the whole process, not nucleic acids. They were the first who could make nucleic acids.
Mm-hmm. Okay, thank you.
Thank you.
All right.
Well, thank you very much,
Yes,
Professor Lupas, for joining us today. And hopefully we will have you at some point in the future
Yes,
for another seminar.
you saw there's now seven vaccines in late phase 3.
So maybe next time you can come to camp
Yeah.
in person.
Yes, I think travel will start next spring.
Hopefully, fingers crossed.
It's obvious, everybody's racing for it. And I saw an incredible revolution in my field, which is going to be made public in a few weeks. Something that's so fundamental, it's going to change the way we operate in biochemistry.